All right. So first part of the day is started. We're gonna have a lecture next. After the lecture, we'll do a discussion on endurance. Depending on how long that takes, I have another lecture up here. But if we don't get to it, it's really easy to push that till tomorrow. All right. That's just a lecture on how to read a scientific paper with an assignment due. If we don't get to it today, the assignment will just be due on Thursday instead of Wednesday. All right. So with that, I don't know if you remember, all the way back to day one a week and a day ago, we looked at space news. All right, and they kind of did their yearly end cap. All right, but I thought we'd do another episode of what's happening this week.
Houston Station on Space to Ground.
Welcome to Space to Ground. I'm Kayla LaFrance. As we say hello to the new year, the International Space Station is setting its sights to many years ahead. NASA Administrator Bill Nelson announced that the Biden-Harris administration is committed to the extension of operations of the space station through 2030. This will enable continuation of groundbreaking research being conducted by NASA and our international partners on the orbiting laboratory through the rest of this decade. In a statement posted to nasa.gov, Nelson said that the International Space Station is a beacon of peaceful international scientific collaboration and for more than 20 years has returned enormous scientific, educational, and technological developments to benefit humanity. One of the current developments aboard the space station is the manufacturing of superalloys. Superalloys are metal alloys with incredible heat-resistant properties. The turbine superalloy casting module is testing a commercial manufacturing device that processes heat-resistant alloy parts in microgravity. The research of these superalloys could improve the performance of materials under extremely high temperatures, such as metals and turbine engines for aerospace and power generators. So how is your New Year's resolution coming along? Well, we have a resolution that will be easy to follow. We invite you to keep up to date on the latest news about the International Space Station this year by connecting with us on one of these social media platforms.
All right, I'm going to pause it there because it's just going to tell you to go like them on Instagram and Facebook and all their other social media platforms out there. Not quite related to plants, but I think it's related to what we'll be talking about today. And let's see, I just forgot on Zoom here that I forgot to do screen sharing. All right, and this lecture is being recorded, so I will post it when we're done here. All right, so that's our space news. Has a little bit of a reference to what we'll be talking about today. Making superalloys in microgravity, because our topic today is on gravity, all right? And specifically, when we're looking at plants, we're dealing with another tropism, all right? So we've looked at phototropism. We've looked at, I guess we've only looked at phototropism. We will look at gravitropism today, all right? So that's growth in response to gravity. One of the tropisms we're not going to look at, but I thought I'd put it up there because it's interesting, has a cool sounding name. All right, is thigmotropism, all right, and that's the response to touch. So a lot of vining plants will have thigmotropism because when they touch something that they can wrap their vines around, all right, they're responding to that touch sense. So we're going to focus on gravitropism today because we're looking at gravity, all right. And so gravitropism, we've kind of learned a little bit about that when we set up our lab, which hopefully everybody has rotated yesterday. If you have not, go ahead and do it today. Make sure you do it today. All right, but you'll have to make special notes on your lab report that you rotated it after 4 days instead of 3. All right, but hopefully everybody did it yesterday. So gravitropism. Plants are orienting themselves in response to gravity. All right, so much like phototropism, they're orienting themselves so that they can find resources. All right, so positive gravitropism, we're going to grow with gravity. All right, so we're going to go down. All right, so positive phototropism, we grew up. All right, we grew with the stimuli. All right, towards the stimuli. All right, gravitropism, we're talking about growing down because we're growing with the stimuli. All right, whereas negative gravitropism is we're growing up. We're growing against gravity. All right, so unlike the other stimuli we've looked at, all right, gravity is always consistent. It is always there. All right, there's no gradient of gravity. All right, gravity isn't more or less depending on where we are on Earth. It does not vary, and we can't turn gravity on and off here on Earth under most circumstances. All right, now I know that NASA has, I think, a tunnel where they can try to turn off and simulate microgravity the best they can. All right, but for most of us here on Earth, we're not going to be able to turn gravity off. Right? You can't switch a switch, and we'll all start floating in the classroom. All right, so it's more than just up and down. All right, so we talked about gravitropism in terms of growing upwards, growing downwards. All right, but it's more than just positive and negative gravitropism. All right, there are parts of plants that will grow horizontally or at different angles. All right, so if we look at diagravitropic, all right, these are referring to plant parts that are going to grow at a right angle to the pole of gravity. All right, so they are growing at 90 degrees. All right, we can see this branch in our image down here. All right, that is diagravitropic. It is growing at 90 degrees from gravity, to the pole of gravity. All right, plagiogravitropic are all those parts of the plants that will grow between 0 and 90 degrees. All right, so a lot of branches, a lot of lateral roots will grow in this direction. All right, so we can see our image is pointing to a lot of these roots here that are growing at kind of an angle. All right, it's not a 90-degree angle, it's somewhere in between. Same with these branches. Right now, if we have parts of the plant that just does not respond to gravity, all right, it's going to grow with little or no regard to gravity. That would be agravitropic. All right, so a good example of agravitropic is some of the lateral roots. All right, once we get branching enough occurring in the roots, some of those branches that are really highly branched are not going to have any regard for gravity. Another good example might be if you've looked at a flower that kind of droops over. All right, that flower part, if it's drooping over, it's not responding to gravity. All right, I guess, we could consider that responding to gravity, couldn't we? It's not growing in response to gravity, but it is responding to the pole of gravity. And again, why respond to gravity? Well, we want those resources, all right? So roots, we need to provide water and nutrients, but roots also need to anchor the plant into the soil, all right? So if it's anchoring the plant in the soil, it has to search below the plant. Stems and leaves, we need to once again acquire resources, just like in phototropism. We need to reach towards the sunlight, all right? Why gravitropism is so important in terms of not just positive and negative, but all the angles in between, is we want to fill up that space. All right, so if we thought about just positive and negative gravitropism, all right, that's all that space there that could have been utilized. All right, so if we add in more than just positive and negative gravitropism, we see that we can utilize a lot more space, we can acquire a lot more resources, we can have leaves not competing with each other for resources like sunlight here. Same with the roots. All right, we're increasing our surface area. All right, so one of the structures that we talk about a lot when we talk about gravity are roots. All right, and this is really where we're going to focus too, because most gravitropism research is focused on root and root anatomy. All right. So if we look at root anatomy, I just want to orient ourselves to what we're looking at when we're looking at the tip of a root. All right, we have these 3 zones, which have been outlined here. All right, we have the area of cell division. Now this contains the apical meristem. So if you remember, meristem is an area of actively dividing cells. All right, so area of cell division refers to apical meristem, actively dividing our cells. All right, next we have this area of elongation. All right, so these cells that have actively divided, they're going to continue to grow downwards and eventually move into this area of elongation, where the cells are going to grow. All right, they are expanding out, they are becoming larger. All right, the last zone, all right, is this area of maturation. I have a really hard time saying that word. All right, but this area is where these cells become mature. All right, they become mature, they're going to differentiate, all right, or they're going to go become the cells that they are destined to be, essentially. All right, so if we need lateral roots, all right, we might have roots that start branching off. Epidermal cells might start to specialize into root hairs. All right, different cells might become specialized to be xylem or phloem in this vascular cylinder and become mature in this area. Now, one of the most important parts when we're talking about gravitropism is actually the root cap. All right, so the root cap is down in this area of cell division. All right, and it's a cluster of these cells that kind of protect the tip of the plant. All right, it protects the meristem. All right, not only does it protect the meristem when these roots are growing down through this coarse soil, right, protecting it from damage. Right, but it's also going to kind of help lubricate the way for downward root growth, all right, because it secretes a mucilage. All right, so it secretes a mucilage that will lubricate the pathway so that once again we can slide our roots easily down into the ground with minimal damage to our meristem. All right, any questions so far? So when we're talking about root gravitropism, we can really think about it as happening in 4 different steps. All right, perception, transduction, transmission, and response. All right, and we're going to go over these steps in a little bit more detail here, but know that this happens very quickly. All right, so actually perceiving gravity takes less than 1 second. All right, responding to that force of gravity is going to take, 1 second to up to 10 minutes, depending on what phase we are. All right. The response is going to be growth, right? And that's just going to be continuous. All right. So that's going to take days upon end, right? But the actual perception of gravity takes less than a second. It happens very quickly. All right. We often think of as plants as very slow moving. All right. So I like to emphasize that this happens fast. All right, so in the first step is perception, right? We are actually perceiving gravity. So in order to understand how plants perceive gravity, right, there's a little bit more anatomy that we have to dive into. So perception is going to happen in the root cap, all right? The root cap protecting the meristem there. And if we look at the root cap, all right, as depicted in this image down here, right, we have these columella cells, which are the central core of cells within the root cap. Right? They're this dark gray in our image here. Right? These cells have the most influence on the perception of gravity in roots. Right? So these play a key role in gravitropism. Now we also have peripheral cells. Right? Peripheral cells are kind of this epidermal layer around the root tip here. Right? They're on the outer edge. It can be 1 or more outer layers of cells. Right? They can play a role in sensing gravity, right? But not as important as a role as these columella cells. All right, now one reason we know that the root cap is where roots are sensing gravity is that if we were to cut the root cap off of a root, right, that plant's no longer going to respond to gravity. All right, so if we cut off the root tip, we turn the plant maybe sideways, all right, that root tip is just going to still continue to grow on the path that it was on. It's not going to respond to any change of gravity. So that's how we know that these cells are super important. All right, also in perception of gravity, all right, we have to have a way to actually sense gravity. All right, so in the root cap, in those specialized cells we talked about, all right, we can say that these cells are statocytes. All right, so statocyte is a term regarding any cell, all right, that contains these starch grains. And these starch grains are held in amyloplasts. All right, so starch grains, they're made up of a carbohydrate. The plants will synthesize these out of excess, glucose from photosynthesis, and that they can store them in these membrane-bound packages called amyloplasts. All right. So when we're binding them in this membrane of amyloplasts, and we can see all of our amyloplasts as these dark dots in our image and our root cap here, right? That makes them heavy, right? So essentially, these amyloplasts are going to fall in the cell according to gravity, all right? So they're going to fall in the cell. Right now we can see that our root tip would be oriented how we would expect a root tip, up and down, all right? Because these amyloplasts have fallen to the bottom of the cell, all right? So that is how the plant is perceiving gravity. All right, well now they have to respond to that gravity. All right, and that first step in responding is called transduction. Now we're not going to go into this in too much detail at all, but what I want you to know is once those amyloplasts fall to one side, all right, the cell will sense that placement of those amyloplasts, all right, and produce signaling molecules. All right, so I put this image up here. It is producing signaling molecules, calcium being one of them, hydrogen ions being another of them. Goes through a series of signaling processes to kind of tell the plant, oh, gravity has changed on us. We have to respond. All right, so after it sends out a signal, all right, the last step in perceiving gravity and responding to it here is transmission. Right? So in transmission, the plant has received those signaling molecules, and that activates auxin. All right, so remember we had auxin in phototropism. We also have auxin acting in gravitropism. All right, so auxin, when these amyloplasts fall to one side of the cell, the auxin is going to be transported to the root, bottom of the root, towards this root tip here, and it's going to fall towards the side, all right, where gravity is telling it to essentially fall. All right, so in this case, our root here is kind of sideways. We have more auxin accumulating on the bottom part of the root, all right, the bottom side of the root. Now, unlike in phototropism, right, so remember phototropism, auxin will tell the plant, the cells on the shaded side to elongate, right? The opposite is happening here, right? So auxin is telling the cells on the bottom portion of the root that is sideways, right, to inhibit elongation. All right, so it's telling these cells don't elongate. That way the cells on the top, they'll grow normally and kind of bend the root downwards. All right, so auxin playing a major role here. So then that last step is response. All right, the actual growth towards gravity. All right, the growth response, it's typically downwards unless we're talking about something that grows on the right degree angle or in between. Right, and we can see this here. We have our amyloplasts. They have fallen to one side, and that is causing our root to turn downwards. All right, so what if we take away gravity? All right, when I thought about this class, I thought, oh, gravity is going to play such an important role in gravitropism on plants. All right, but then I did some more research, and not so much. All right, we take away gravity, the plant's going to rely on phototropism instead. All right, so instead of sensing up and down, it's just going to sense where the light source is. All right, roots are in the dirt. The dirt is taking away any light. All right, so the roots are perceiving darkness instead of gravity. All right, now if we look at a plant that's been grown without any gravity, right, shoots tend to be more upright, right, because gravity is not there to allow them to droop over. So they kind of have a weird look to them, all right, because we have no gravity causing the leaves to droop. But as some of you might have picked up on by now, all right, those articles I had you read were talking about auxin and how auxin is not as regulated by gravity as we thought, all right? So up until this point, all the textbooks were like, yes, auxin. Auxin is the major key player in causing gravitropism. If we take away, gravity, then there should be some sort of a response with oxygen. But we're finding that's not the case. All right. Now, I don't really know why. All right. We're still researching that, but it's leading to some interesting discoveries in plant biology. All right. So I have another fun video here. It's talking about plants in space.
I don't know about you, but I never eat vegetables. They're just so... But maybe I'd be more tempted to eat them if they came from outer space. Hey guys, Julia here for D-News. So recently, astronauts aboard the International Space Station chowed down on the first bite of space lettuce. It's technically red lettuce, but they call it outrageous lettuce. Get it? Get it? Oh, it's so bad, it's good. Puns aside, I'm super stoked that NASA astronauts Jell Lindgren and Scott Kelly, along with Japanese astronaut Kinya Yui, got their dose of leafy greens while orbiting 220 miles above the surface of the Earth. In what's considered an important step to self-sustaining systems, NASA scientists grew the lettuce in microgravity over a period of 35 days. It's not the first batch, though. Other plants have been grown aboard the station. One study published in the journal BMC Plant Biology discussed small white flowers that grew in space to see how well they would grow without a lot of gravity. And just last year, lettuce was grown that was sent back to Earth to be analyzed for safety concerns. Once they got the A-OK, another round of plants were grown. So how do you grow plants in space? It's a little tricky since plants have evolved stems to fight against gravitational forces, so how would they grow in low and microgravity? Well, pretty well apparently. ISS uses the veggie system, developed by Orbital Technologies in Madison, Wisconsin. The system contains lighting and nutrients for a space garden. Yes, space garden. This particular garden contains red, blue, and green LEDs. The red and blue mostly for plant growth and health, the green so the plants don't look weird under the red and blue lights and more resemble lettuce grown here on Earth. The plants get their nutrients from a pillow. The pillows can come in various sizes depending on what kind of veggies are grown in them. Pillows only work once though, but that's by design. That way there's a reduced chance of microbial growth from the system of plants. For plants that might have more microbes, a sanitation method they can do in the space station will have to be developed. Like Scott Kelly and his buddies, for instance, use citric acid-based food-safe wipes to clean the lettuce before eating it. Besides providing a source of nutrients that's not freeze-dried, maybe space gardens can offer other benefits too. Dr. Joya Masso, one of the scientists working on veggie, says there may be a psychological benefit. In extended manned missions with only a half a dozen or so crew, space can be tiring and lonely. But tending to a garden might give the astronauts mood a boost. Some studies, like one published in the journal Science, show that gardening and greenery increases positive mood. Gardening helps people feel more relaxed, pleased, and optimistic. Definitely feelings we might need as we leave our home planet. Growing plants without Earth soil will be crucial to any long-term trip to a planet like Mars. With limited resources and not a lot of chances for restocking, expeditions will have to be self-sustaining. Scott Kelly called this experiment a big step in that direction. But this research can help us here at home too. A lot of the same technology, the LEDs, the pillows, and the watering system can help us farm more efficiently. We can grow food in smaller spaces and with less energy. So cool space beans! And just in case you were wondering, Scott Kelly said the lettuce tasted awesome. Speaking of awesome innovations back here on Earth, Intel creates the breakthrough technologies that make amazing experiences.
All right, we've come to the self-promotion part of the video here. But I want to back up when she talked about growing plants in space and using these pillows. All right, so if we're thinking about gravity, if we had a handful of dirt and we released that in the space station, that dirt's going to go everywhere. All right, so instead of having open-ended pots like we do here on Earth, they had to come up with a way to grow plants with soil and not have that soil go everywhere. All right, so these pillows are one way that they do that. All right, it's essentially this self-contained system, right, with just a small opening where the plants can grow out of. All right, that way it doesn't create a big mess. All right. All right, so let's take a break and think about what we've learned. All right, so I want you to just revisit. What is the root cap? What does it do? All right, and what are those 4 steps in gravitropism? And while you do that, I'm going to pass back the drawings that you made the other day.
I'm so sorry. Satisfying.
All right, so if you completed the drawing, all right, I gave you 2 points. It looked like everyone did a great job. One of the things I didn't notice was that some of the drawings were missing carbon dioxide and the Kelvin cycle. Now, I did not require you to do the Kelvin cycle, but I did ask that you put carbon dioxide in there, which goes into the Kelvin cycle. All right, so I didn't count it against you if you forgot that, but I just wanted to make that note. All right, what is the root cap? If I was looking at a root, where would I find it? The very tip of the root, yes. And what does it consist of? It's 2 different types of cells. One starts with a C, one starts with a P. Yes, columella and peripheral cells, all right? So peripheral cells are on the outside. The columella cells are the central core of cells in that root cap. All right, so what does the root cap do? It has 3 different functions that I talked about. Protects, lubricates, and the big one that we spent multiple slides looking at, senses gravity. Yes, yes. So it's not only protecting the meristem, all right, and providing lubrication through the soil, right, but it's sensing gravity as well. All right, so what are those 4 steps in root gravitropism? Perception, yes. What's happening in perception? We're perceiving something. We're perceiving gravity, yes. All right, what is the next step? Transduction, and what happens in transduction? Yeah, the cells have sensed that those amyloplasts have fallen, and they are producing a signaling molecule. All right, next step. Transmission, and what hormone plays a huge role here? Auxin, yes. And what does auxin cause the bottom side of a root to do? Stop growing, yeah. It's inhibiting elongation of those cells. All right, and the last step would be response, where the plant does what? Grows, yes, very nice. All right. All right, not sure why my screen sharing isn't working here. But I did record it and put it on Moodle here. All right, so one of the things our last video kind of led us into talking about was pathogens, right? Microbes. So they mentioned that Scott Kelly, right, kind of, when he went to eat the lettuce, wiped it down with these, it sounded like antimicrobial wipes to kind of clean off any microbes that might be at the surface. Right, so in space, pathogens are of a huge concern. Right, we don't want anything contaminating our food or our astronauts. Now, it is rare for pathogens to be transferred between plants and humans, right, but it can happen in some instances. All right, so when I'm talking about pathogens, I'm talking about microbes. All right, small organisms, often single-celled, but not necessarily. All right, so bacteria, fungi, viruses, those would all be microbes, right? They're all disease-causing, right? So if they infect the plant, they're going to cause some sort of disease in the plant. And the way that they do this is by producing a molecule called elicitors, all right? So these elicitors are compounds that are going to kind of promote the infection of these plant cells, all right? So essentially, these microbes are coming in, they're producing these elicitors, and moving their elicitors into the plant cells to cause some sort of disease to happen. All right, so plants need to defend themselves. All right, I threw up an image here, all right, of just different types of diseases that plants can get depending on what microbe infects them. All right, it's a huge range of diseases that can be caused. All right. In general, it all kind of happens in the same way with these elicitors and then plants responding in some fashion. All right, so plants, they have to defend themselves against pathogens. All right, and they have several different strategies to do this. All right, we're going to talk about a couple of these strategies. On their plasma membrane, all right, so if you remember, the cells are lined with a plasma membrane on the outside of a cell, all right? They're going to have receptors, these molecules that can sense different molecules in the space surrounding the cell. Right? So these receptors can bind to very specific microbial molecules. Right? So it can bind to those molecules and help stop an infection from occurring. All right, now they might have what's called microRNA. All right? Think of it as a type of DNA. We're not going to get into too much detail into microRNA here. But it's in the cytosol, all right? So the cytosol is the cell fluid inside of the cell, all right? So we know we have a nucleus, we have organelles inside of the cell. The space surrounding all those organelles are fluid-filled, all right? That is the cytosol, all right? These microRNAs are going to essentially destroy the nucleic acids of invading viruses, right? So nucleic acids are kind of the building blocks of the molecules that these viruses are made up of. All right, so we're going to destroy those building blocks, all right, thereby helping prevent infection. All right, there can also be receptors inside of the cytosol, right? Remember, that's the cell fluid that can recognize those elicitors that have been injected into the cell by those microbes and trigger the production of a chemical defense. All right, we can trigger some sort of molecule to be produced that's going to help defend against those elicitors. Or that cell might simply have tell itself to go through a programmed cell death, right? So if we kill the infected cell, hopefully it won't spread to other cells, right? That's the idea behind programmed cell death. All right, so one of the responses we'll talk about is this hypersensitive response, all right? Now this occurs when a plant recognizes a pathogen and responds so that those symptoms will be limited, all right? So one of the ways that it can do this is by increasing its production of hydrogen peroxide, right, as well as another molecule called nitric oxide, all right? we'll see how that works in just a bit here. They can also produce other secondary metabolites that might help defend themselves, right? So we're producing compounds in response to these elicitors. All right, now I threw up this image here because I think this image really helps us understand what exactly is happening. All right, so at the top here we have our pathogen, all right, two different pathogens actually, that are producing elicitors. All right, notice different pathogens can produce different elicitors here. Right, but they're being, one of them is being injected into the cell, right, which we don't want to have happen, right? The other one is being stopped by one of those receptors we talked about, right? It's immediately intercepting those elicitors and not allowing it to go into the cell, right? But if it does enter the cell, all right, we can produce more hydrogen peroxide, this H2O2, right? This H2O2 can help kill pathogens. It can also help strengthen our cell walls to try to prevent these elicitors from getting through the cell walls any further. Right? Now it can also produce this nitric oxide compound, and together with hydrogen peroxide, it's going to create other defense compounds and hormones that can do a variety of things. All right? It can send signals to other parts of the plants to tell it, the other parts that I'm being attacked, beware, start preparing. All right? Some of these defense compounds might be, we're talking specifically about pathogens, but the same thing can essentially happen with herbivores. So say we have a bug chewing on the leaf that might produce chemicals that are bad tasting. All right? Oftentimes when we're talking about pathogens, though, we're talking about alarm signals. Now it can also lead to cell death. And we can see that in our image here. All right? We have these spots. We call them necrotic spots here. Right, where the cells have essentially just died. Right? They're dying off to try to stop the spread of the infection. All right, so that's a very localized response to pathogens. Right? But we can also have a whole plant response to pathogens. Right? So this would be systemic acquired resistance. Right? So this can result in that production of those alarm signals that we were just talking about that can alert the whole plant to this invasion of a certain microbe. All right, it induces a very widespread resistance. And one of the compounds that a plant might release in response to this widespread resistance is jasmonic acid. Right? Now, I'm sure all of you have probably smelled jasmonic acid before, right? Because this is a compound that when you're mowing your lawn, these plants will send out. So that smell of freshly mowed grass is actually this jasmonic acid, right? It's a signaling molecule telling not only other parts of the plant, but other plants of the same species that, hey, something is attacking me, be prepared. All right, now we might also produce other defensive enzymes that might be toxic to those microbes and kill off those microbes. All right, but again, systemic is the whole plant response. Hypersensitive is just a very localized response. All right, well, how does a plant know when to defend itself? All right, so plants need to defend themselves, but plants don't always need to constantly be defending themselves. All right, so we can break down their defenses into two different categories. So one would be a constitutive defense. All right, so this is a constant defense. This defense is always on, it's always defending the plant. All right, so this might be in terms of really thick cell walls. Maybe we have a thick cuticle on top of our leaves to prevent microbes from even getting through. Bark is one of these responses. These defenses that are always on. Right? Bark is hard and thick and tough to get through. All right, so a constitutive defense, always on. All right, but now some of these defenses are very costly to synthesize and make. All right, so these would be an induced defense. All right, so an induced defense is a defense that is only created when it is needed. All right, so essentially it's only created when the plant senses that it is being attacked. All right, because it's costly to make. We don't have to constantly want to make these molecules that are using up all of our resources if we don't need them. All right, so these resources might be toxic chemicals. All right, it might be enzymes that help to degrade pathogens. All right. It could be deliberate cell suicide, right? That programmed cell death that we talked about, right? Those would be induced defenses, only created when they are needed. All right, so this video might be a little bit of a stretch in relating to our topic today, but I thought it was really interesting how working with NASA has actually benefited wineries.
It's impossible.
Ooh, let's back
The
that up.
real As farmers and winemakers, we work very hard to grow the best quality fruit possible to make the best wines possible. The real polished part of winemaking is letting that wine age gracefully so those flavors can be preserved and brought to your home into enjoying the glass. The greatest issues we face as winemakers is that our optimal storage conditions for our wines are also optimal growth conditions for mold and bacteria.
Mold is really bad in a barrel room or in a cellar because it has an odor to it. And the wines are a living, breathing thing. And actually, they pick up the characters of where they're living.
The wines can absorb that aroma, and it's just not a healthy environment to work in. My father and I built this barrel room underground. The challenge is we've got some stagnant air which can cause mold growth. Who knew that our solution would be not underground, but up in the sky?
In order to carry astronauts to Mars for deep space exploration, you have to be able to grow your food storage. You don't have enough space and capacity to carry them with you. So NASA began experimenting with growing crops in space. What they found is that in the growing chambers on the International Space Station, the crops they were growing were emitting a naturally occurring hormone called ethylene. It's a gas, and ethylene is the reason that fruits and vegetables ripen.
But the problem was in space we're going to have a very confined environment, so the ethylene concentration can build up, especially in a plant growth chamber. It basically destroys the plant. And so there was a need to grow plants in space, and we were good at growing plants here at the University of Wisconsin, and so NASA came to us and said, we've got a problem, I think you've got a solution, can you help us?
Through the University of Wisconsin Advanced Astroculture Program, they developed an ethylene removal system that was successfully tested on a number of missions on the International Space Station. So the ethylene removal system that was developed draws air through a reaction chamber. The air is passed across the catalyst bed, and that catalyst has the effect of removing anything organic from the air. That includes mycotoxins, bacteria, viruses, and even mold.
It showed us that with the right technology, we can overcome the challenges of growing plants in space and enable future space explorers to grow their own food as they continue to explore the solar system.
The International Space Station program not only supports the technology that allowed us to get rid of ethylene in the Space Station Astroculture program, but it also provides a great variety of technologies to exist that otherwise wouldn't have been possible.
Realizing that this technology could have tremendous benefits here on Earth, we developed the Aeroside air purification systems. Initially used in the perishables world for prolonging the shelf life of fruits and vegetables, not long after that, we realized because of the effects the technology has on mold, that the wine industry was a perfect complement for the technology.
This technology has been a real boon to us. It helps me sleep better at night. But as you can see around this other, there's not a lot of mold.
Within 24 hours, the amount of airborne mold spores were dramatically reduced.
Within 2 weeks, about 99.9% of the mold had vanished on the walls.
It solves problems before they occur.
The room smelled fresher. It actually enhances your storage conditions for you to even make better wine.
It's really amazing when you think about all the innovations that are going on up in space, how they can come into a place as unexpected as a winery, which translates into benefits on your dinner table.
Alright, so I like this video because it kind of links back to pathogens when we're talking about bacteria, right? But it really helps us understand how these crazy expensive experiments in space that are kind of designed for space living can be translated to help us back on Earth. Alright, so it wasn't intended as a product used in the wine industry, but as a result of being really good at removing bacteria and molds, it was perfect for the wine industry. So I thought that was a really interesting connection there. All right, so that's the end of the lecture part of today. All right, so the homework is to watch a couple videos here. I don't know that we're going to get to the scientific paper assignment. All right, if we don't get to it, you can ignore that half there. We're going to discuss endurance in, let's say, 5 minutes here. Get up, walk around, use the bathroom, whatever you need to do. And then the next part of endurance we'll discuss next week. So start thinking ahead, chapters 6 through 13. All right. So go ahead, take a break, jump around, get ready to discuss. Yeah.
So for the class next Monday on MLK Day,
Yes.
we're starting at 11:30.
Yes.
Do you expect it to go all the way till 2 o'clock?
What do I have planned for that day? Let's look at the schedule. Let's see. Yeah, next Monday is Martin Luther King Day, and there's a virtual presentation. So all classes have been pushed back an hour, which means we could end at 2 instead of 1. I don't envision us going that long. No.
I'm scheduled to work at 1:15.
All right.
Is that, like, because I can, get that worked out if we're still in class, but I'm just wondering if I should.
That's okay. I think we should be fine by then. You work
Okay.
on campus? Or?
Yeah.
Okay,
Just
yeah.
on campus.
Yeah.
Okay.
You'll be fine.